So we'll start our first session with Professor Dr. Wang. So unfortunately, I think Professor Dr. Lingle Wang is away and he has sent us a recording. So we'll start with the recording of the session. And in case if he's there towards the end of the talk, we will take up the discussion session or we'll write to him for the discussion. Fine. I hope that is fine, Nishan. Okay, all good. So shall we go ahead with the
thank recordings? You. Yes. Yes, British.
First, thank the organizers for arranging the conference and for inviting me to speak here. I'm happy to present some recent work at Schrödinger for protein ligand binding field calculations. Schrödinger is a scientific leader in computing chemistry, providing software solutions and service to advance drug discovery projects. We are particularly interested in any computing method that has potential to transform drug discovery projects. Today, I'm going to talk about our methods for protein ligand binding field calculations. A little bit background about why we are interested in this problem. As we all know, drug molecules achieve their therapeutic effects through binding to the protein binding pocket, either blocking the protein function or changing their conformations to alter the downstream single transduction pathway. In either case, the efficacy of drugs are correlated with their binding affinity. As a result, the major objective of small molecule lead optimization, one of the most expensive stages in drug discovery projects, is to identify molecules with improved binding potency while maintaining a list of other ligand properties to achieve the safety and the biological efficacy. Therefore, accurate calculation of protein binding affinity is of key importance in computational drug discovery. Atomics model affinity derivation is the most rigorous method to calculate protein ligand binding affinities. At Schrödinger, we initially were focused on relative binding affinity calculations between catenaric ligands, following the summary cycle shown here. FEP is a rigorous theory, but the accuracy is critically dependent on two aspects. First, we need an accurate force field to describe the atomic level interactions between proteins, ligands, and water molecules. Second, we need an efficient sampling method so that the relevant and the important conformation space for the two physical endpoints can be finally sampled in a relatively short MD estimations. For large biomolecules like proteins with many local minima separated by a high energy barrier, using brute force MD estimations, the system will stay trapped in the local minima corresponding to the initial geometry and configuration. And the calculated binding affinity using brute force MD will be systematically dependent and biased towards the initial configuration, introducing large errors in the calculated binding affinity results. And at Schrödinger, we are addressing both the force field accuracy and the sampling efficiency problems. With over a decade of efforts, we have developed by far the most accurate force field, OPS4, for small molecules and proteins. And we are continuing our effort to further improve the accuracy for the force field. At the same time, we have worked out a number of methodological improvements that dramatically improve the efficiency to sample the relevant conformation space associated with ligand binding, including the ligand and protein cytosine conformation changes and water relaxation in the deeply buried binding pocket. Combining those efforts, we have developed by far the most accurate assay F+ for protein ligand binding affinity calculations, which has been used as a benchmark for other methods development. In the initial paper published in 2015, we validated F+ method on a set of 8 pharmaceutical interesting drug targets covering a diverse chemical space with more than 200 unique ligands. And the calculated binding affinity are highly correlated with experiment, with overall root mean square error of about 1.1 kcal/mol, and the errors are roughly Gaussian distributed. After that, we have introduced a number of novel methods that dramatically improve the accuracy and expanded application domain of affinity calculations, enabling any complex and diverse perturbations to be modeled with very high accuracy, including scanfold or co-hopping modifications, macrocycle perturbations, charge perturbations, reversible and irreversible covalent inhibitions, and so on. And at the same time, with the continuous improvement in the force field accuracy, the reliability and accuracy of calculations has also been further improved. In the next few minutes, I'm going to present and focus on a few novel methods that dramatically expanded the applicant domain and improved accuracy of fairness calculations. Historically, FEP method was designed for relatively small perturbations, for example, mutating the methyl group into a hydroxyl group, or adding a fluorine atom on an aromatic ring, or changing the nitrogen linker in the molecule into an oxy linker. Accurate or reliable calculation for those type of perturbations already allows a large number of molecules to be tested. And as we saw already, it already generated a large impact in real discovery projects. However, the impact of FEP will be much limited if we restrict ourselves to those kind of very minor and modest modifications. In drug discovery projects, chemists would like to explore a much diverse chemical space and very often consider very challenging positions. For example, if the core of the molecule has certain atomic toxicity issues, they may want to introduce additional ring or to change the size of the ring in the core of the molecule. Those are so-called scaffold hopping or co-hopping modifications. So how to calculate the affinity change for a co-hopping modification? Another example, for targets that do not have deep binding pockets, blocking the protein-protein interface is a very viable strategy. And the macrocycles are very good candidates to block protein-protein interface. And how to calculate the affinity change to cyclize a linear molecule into a macrocycle. Those type of relations are very challenging for conventional method. And in the following, I'm going to explain the unique challenges associated with those conditions and our methods to address those. So co-hopping, as mentioned earlier, in drug discovery projects, chemists often would like to consider conditions that change the size of the ring or introduce additional rings in the middle part, in the core part of the molecule. Those type of conditions are called scaffold hopping or co-hopping modifications. Well, it might be relatively straightforward to revise the synthetic route to make a molecule with a different R group. Synthesizing a molecule with a new core may require months of chemistry effort. And since the topology of the pharmacophore is modified in a co-hopping modification, the majority of co-hopping attempts failed to provide any valuable matter to the project because the majority of co-hopping attempts resulted in a molecule that is not active. But even that, co-hopping is often attempted in drug discovery projects because very often it is the only viable strategy to resolve a particular enemy toxicity issue. And also the novel IP generated by co-hopping makes it even more attractive. So therefore, accurate calculation of the affinity change for co-hopping modification will enable the chemists to pursue co-hopping modifications with confidence that the resulting molecule is potent enough and useful for the project. So co-hopping is a novel topic with no earlier research on this area before. And here I will use one example to explain the fundamental difference between the conventional R group modification FEP versus the co-hopping FEP. For R group modification FEP, for the example showing here, mutating from the chlorine atom into a hydrogen atom, if we retain all the leftover, the bounded stretch interaction between all the leftover dummy atom and the rest of the molecule, we can easily see that the ensemble of configurations for the mixed topology molecule is the same as for the target physical molecule. In other words, the bounded stretch interactions between leftover dummy atom and the rest of the molecule won't affect the ensemble of configurations. So therefore, we do not need to calculate the affinity to form or to break a covalent bond. But this is not the case for co-hopping FEP. For the example showing here, to mutate from a fused ring into a linear molecule, if we retain all the bounded stretch interaction between the leftover dummy atom and the rest of the molecule, we can easily see that the ensemble of configurations sampled for the mixed topology molecule with the dummy atom and the bounded stretch interaction will be different than the real physical target molecule. For the physical molecule without the dummy atoms, it can assess many different conformations through rotation around this two rotatable bond. By comparison with the dummy atom and the bonded stretch interactions, the ensemble of configurations will be much limited. So therefore, in co-hopping FPP, we have to explicitly calculate the finish to form or to break a covalent bond, which was believed to be extremely difficult or even impossible. There are two unique challenges associated with the calculation of the finish to form or to break a covalent bond. The first one is singularity, numerical instability problems. So in almost all the commonly used molecular mechanics force field, the bonded stretch interactions are represented by a harmonic potential. And using the conventional FEP to scale the potential by a coupling parameter lambda, we can easily see that the derivative of the finish with respect to the coupling parameter lambda diverges and is undefined when lambda approaches zero, introducing singularity and numerical instability problems in the finish calculations. The second challenge is associated with the convergence of the finish. So when lambda equals 1, with the bounded stretch interaction fully turned on, the distance between two atoms approximately follow a Gaussian distribution, approaching delta distribution with a very strong force constant. By comparison, when lambda equals 0, when the bounded stretch interaction is turned off, the distance between the two atoms approximately follows a flat distribution. Those two distributions differ so much that it has very, very little phase force overlap. And it's very difficult to design an intermediate state that has sufficient phase space overlap with both physical endpoints. And to address those challenges, we have invented a novel functional form, which we call the soft bond potential. The formula is showing here. We can see that when lambda equals 1, this equals 0. So this potential recovers the harmonic potential used for the bounded stretch interaction in conventional force field. When lambda equals 0, this potential goes to 0. And for lambda values between 0 and 1, the potential changes slowly from harmonic to 0. Both the first and second derivative of the potential with respect to lambda and R are continuous and valid. And through rigorous statistical mechanical derivations, we prove that using this potential, it solves the singularity and numerical instability problems associated with the calculation of the finish to change, to form, or to break a covalent bond, and also resolve the convergence issues in the calculations. The soft bond potential for co-hopping FEP implemented in FEP tasks allows reliable and accurate calculation of the finish for co-hopping modifications. So here's one example. Receptor is EZH2. The company Epsilon just has the first drug approved for this target. The 4 molecules shown here are close analogs of the drug molecule. So for molecules 22 and 27, it has a fused ring in its parallel molecule. For the corresponding molecule 29 and 31, the fused ring is opened to form a single ring. For the perturbation, to open the fused ring into the single ring for 22 to 29, and also from 27 to 31, the co-hopping FEP calculated binding finish is in very good agreement with the experiment. One important property and interesting for the system is the strong line activity effect. In particular, with the 5-member ring in the tail of the molecule, opening the fused ring into the single ring only modestly increased binding potency by about 0.5 kmol. By comparison, with the 6-member ring in the tail of the molecule, the same perturbation to open the fused ring into the open ring dramatically improved the binding potency by about 2 kmol. And the strong line additivity effect is also accurately captured by co-hopping FEP. Another example, receptor space 1. For the perturbation, they introduce an additional ring to mutate from the fused ring into the bridged ring. The co-hopping FEP calculated binding finish is also in very good agreement with experiment. And in this paper, we have validated co-hopping FEP on a set of 6 pharmaceutical imaging drug targets covering many different co-hopping perturbations with diverse chemical space. Again, the co-hopping FEP calculated binding finish is in very good agreement with experiment with overall RMC of about 0.5 kmol, R squared is 0.81. Very recently, we have also extended the same soft bond potential to address another type of very challenging perturbations, the macrocycle perturbation. Macrocycles occupy a unique region in chemical space, bridging between small molecules and large biomolecules. Macrocycles are usually very difficult to synthesize. However, due to their unique ability to balance many different drug properties, including potency, selectivity, membrane permeability, and bioavailability, macrocycles are often considered in drug discovery projects, particularly for targets that are difficult to drug by small molecules. In macrocycle drug discovery, there are two most frequently asked questions. The first one is whether a macrocyclization strategy to cyclize a linear molecule into a macrocycle will improve the binding potency. Second one is, what is the optimal linker size and chemical matter for cyclization? Both perturbations are very challenging for conventional FEP method. And using the same soft bond potential and adjustment parameters specific for macrocycles, FE plus can accurately evaluate both perturbations. And in this paper, we have validated the macrocycle FEP on a set of 7 drugs, interesting drug targets covering many different macrocycle perturbations with diverse chemical space. And again, the calculated binding affinity is highly correlated with experiment with our RMC of about 1 kmol, R squared is 0.7. The accuracy of F plus calculations has been independently validated by a large number of pharmaceutical and biotech companies. For example, Bayer has applied F plus on their internal very challenging data set, including perturbations that cyclize a linear molecule into macrocycle. And the calculated binding affinity, again, is in very good agreement with the experiment, with the results published in this paper. Combining all those improvements, H plus allows very complex and diverse molecules to be modeled with very high accuracy. For example, for a set of HP90 inhibitors shown here, from ligand 69 to 65, the charge in the side chain is removed and the different charge was introduced in the macrocycle, and the overall effect is accurately calculated by F plus. Another example, from ligand 65 to 68, a ring was introduced in the side chain, and also the size of macrocycle is modified. Again, the overall effect is accurately calculated by F plus. In 2017, we spent another effort and collected F plus performance on about 90 projects that we were able to have access to. The majority of them are prospective studies. Some come from our customers. And the overall performance is showing here. The mean unsigned error for the aggregated data is less than 1 kmol. For the majority of projects, for more than 80%, the mean signed error is less than 1 log unit. In agreement with the accuracy seen in retrospective validation. However, we can also see that for a very small percent of projects shown here, the mean signed error is approaching about 2 kmol, much less accurate than other systems, and also making it not very useful for small molecule need optimization. So what are the reasons for those outliers? After a careful investigation of those outlier cases, we have identified 3 areas that can further improve the accuracy and robustness of protein binding affinity calculations. A, improving the sampling of water relaxation in the deeply buried binding pocket. For example, for the HP90 system shown here, there are 2 water molecules in this deeply buried binding pocket. For perturbation, they introduce different chemical group to displace those 2 water molecules. If the perturbation was initiated without the water in the binding pocket, there will be an artificial dry region in the binding pocket, even in the presence of a smaller ligand. If FEP was initiated with those 2 water molecules in the binding pocket, the water molecule will remain trapped in the binding pocket, even in the presence of bigger ligand. In either case, large errors will be introduced in FEP. And by improving the sampling of water relaxation in the deeply buried binding pocket, the accuracy of FEP can be dramatically improved. B, rigorous sampling of the pronation and tautomeric state of ligand and protein residues in the binding pocket. For many systems we are interested in, like the example shown here for the base systems, the pronation and tautomeric state of ligand is coupled with that of the residues in the binding pocket. And a large portion of FEP plus outlier cases are due to improper assignment in the pronation and tautomeric state in the ligand or protein residues in the binding pocket, or failure to identify the change of pronation state associated with ligand binding. So therefore, by rigorously modeling the pronation and tautomeric state of the titratable residues and ligands in the binding pocket, FEP plus accuracy can be dramatically improved. C, improve the sampling of ligand-induced protein conformation changes. For example, for the HP90 system shown here, in the presence of smaller ligand, this part of protein is adopting a looping conformation pointing towards the ligand with a very small R group extension. Adding a 5-member ring in this part of ligand, it pushes the looping to adopt a helical conformation. The looping to the helical conformation protein cannot be sampled in a very short distillation, causing large errors in FEP plus results. Therefore, by improving the sampling of ligand-induced protein conformation changes, the accuracy of French calculations can be dramatically improved. And over the past few years, we have been working on a number of novel methods that address those challenges and further improve the accuracy and robustness of protein ligand binding French calculations. For example, to improve the sampling of the water relaxation in deep barrier binding pocket, we have implemented the ground canonical Monte Carlo GCMC method to enhance the sampling of water relaxation. The GCMC enabled FEP dramatically improve the protein ligand binding French calculations. So for a large aggregate of dataset covering many different protein targets, focusing on perturbations that displace water molecules in the binding pocket, the GCMC-enabled FEP got much improved results with errors much smaller than the conventional FEP. To improve the sampling of ligand and the protein residue pronation tautomeric state, we have been working on a general constant pH method. And the constant pH method calculated protein PK results are in very good agreement with experiment. And we are in the process of integrating the constant pH into protein ligand binding French calculations. And to improve the ligand-induced protein conformation changes, we have been working on a novel method that can accurately calculate the phase difference between the different conformations of the protein. For the HP90 example I mentioned earlier, for the perturbation that introduce additional R group, using the looping conformation of protein, the bigger ligand has severe steric clash with the looping conformation and splitting crest. By comparison, using the helical conformation of protein, the bigger ligand was predicted to bind by about 6 kmol stronger than the smaller ligand, a severe overestimation as compared to experiment of -3.2. Using protein conformation FEP, the looping conformation was predicted to be favored by about 2.6 kmol in the presence of a smaller ligand. And combining protein conformation FEP and ligand preparation, the bigger ligand was predicted to bind by about 3.4 kmol stronger than the smaller ligand, in very good agreement with experiment. And we are in the process of automating this process and potentially release this in our future version of the product. In addition to relative binding French calculations with the main use case in small molecule lead optimization, we have also been working on absolute protein ligand binding French calculations to improve the hit rate in virtual screen. So as we all know, for any drug discovery projects, after the target has been selected, the first step is to explore a diverse chemical space to identify as many novel hits as possible. Without the initial hit and lead, we cannot use relative binding affinity FEP to improve the ligand properties. As a current best practices, empirical scoring function is used in virtual screening to dock a large number of compounds And due to the limited accuracy in empirical scoring function, the average hit rate is only about 1%. So therefore, the project team needs to purchase a large number of compounds to identify a few hits. For molecules that are not readily purchasable, making them can be very expensive and time-consuming. And even for compounds that are readily purchasable, setting up the biological assay to confirm their biological activity can sometimes be very expensive and time-consuming. So therefore, any methods that can improve the hit rate is very valuable. And for that purpose, we have worked out absolute protein binding affinity, the so-called ABFEP, to enable the scoring of molecules with diverse scan folds in different chemical space. And by rescoring the top compounds in work screening by ABFEP, the hit rate can be dramatically improved, as you will see from the later discussion. So the thermodynamic cycle for absolute binding FEP is shown here. So we need two explanations. In the solvent neck, starting from the physical ligand, interactions between ligand and the water is slowly turned off to become a dummy ligand. For the complex neck, starting from the dummy ligand with a set of restraints to put the ligand in the binding pocket, interactions between the ligand and the environment is slowly turned on, followed by relaxing the restraint potentials. The purpose of the restraint potential is to keep the relative position and orientation of ligand with respect to protein binding pocket to prevent it from drifting away from the binding pocket in intermediate long windows. And the final difference between the unrestrained dummy ligand and the restrained dummy ligand in the complex is calculated by an analytical formula. We have validated our ABFE method on a large number of systems, including small fragments binding to different protein targets, the entire 8 systems from the JAX publication, and also the respective work screen dataset, which I will discuss in detail later. The performance on the 8 systems from the JAX publication is shown here. The ABFEP average, the overall RMC is about 1.11 kcal/mol, and the overall RMC average R squared is 0.61 kcal/mol, both comparable to the relative binding affinity FEP results. In addition to the JAX dataset, we have also validated ABFEP in the context of hit discovery. Here's one example. Receptor is pseudokinase domain of JAK2 receptor. Jorgensen group performed virtual screening on about 3.8 million drug-like compounds from Zinc 15 database using GLIDE. Then from the top 1,000 compounds based on visual inspection of the binding pose, rescoring with XP, Gold, and MMGSA, they have identified 30 compounds to be purchased and tested. By eye, those 30 compounds have very good binding pose, very good interactions with the protein binding pocket. They have comparable or even better docking scores than the 13 known JAK2 inhibitors. Therefore, they have very high confidence that at least some of those compounds will bind to JAK2, so they expect to discover some novel hits from this exercise. Of those 30 compounds, 27 were successfully assayed and tested, and to their very surprise, none of them showed detectable binding to JAK2. So it's a completely failure for virtual screening. But this is a very good test set to test ABFEP's ability to improve the hit rate and to differentiate actives from inactives. So we run ABFEP on those 40 compounds, including the 13 known JAK2 inhibitors and 27 transient decoys. Here are the results. Using the top poses from GLIDE as initial pose for ABFEP, the enrichment curve from ABFEP score versus GLIDE is shown here. As mentioned earlier, GLIDE score cannot differentiate the actives from inactives. The majority of the inactives has even better docking score than the actives. Therefore, the AUC for GLIDE is very poor, 0.03 and 0.11 respectively. By comparison, ABFEP can clearly differentiate the actives from inactives. The AUC is improved 0.8. In Jorgensen's paper, they also performed very expensive metadynamic explanations to pull the ligand from the binding pocket. And the stability of the ligand from the metadynamic explanations was shown to be able to better differentiate actives from inactives. The ABFEP performance is even better than metadynamics. So for some of the known JAK2 inhibitors, the crystal structure is available. So we also tested the sensitivity of results with respect to the initial binding poses for ligands. For ABFEP, the better ligand poses translated into better accuracy, and the AUC is improved 0.95, almost perfect. The actives stand out from those ABFEP populations. By comparison, for other methods, the results are not very sensitive to ligand pose. So for those 13 known inhibitors with experimental binding affinity available, we can also compare the ABFEP results with experiment. ABFEP results are highly correlated with experimental binding affinity for those known JAK2 inhibitors. Using the top poses from GLIDE, the R-squared is 0.41, while using the crystal poses when available, the R-squared improved to about 0.7. So here's a summary of my presentation. So with many years basic research in force field and enhanced sampling methods development, we have developed the F+ method that can accurately and reliably calculate protein binding affinity and enable very accurate and reliable calculations for very challenging populations, including co-hopping or Stanford hopping modifications, macrocycle populations, co-1 inhibition, and also the charge populations from the benzene PP, which I didn't mention in this presentation. And both retrospective testing and prospective performance has demonstrated the high level of accuracy in F+ calculations for protein binding affinity. In addition, through a careful analysis of a large aggregate of F+ data, we have identified 3 areas that can further improve the accuracy of protein binding affinity calculations. A, improving the sampling of water relaxation in the binding pocket. B, rigorous sampling of ligand and protein residue formulation tautomeric states. C, improving the sampling of ligand-induced protein conformation changes. And over the past few years, we have been working on a number of novel methods to address those challenges. For example, we have implemented the ground canonical Monte Carlo, the GCMC method, to improve the sampling of water relaxation in the deep buried binding pocket. And GCMC-enabled FEP dramatically improved the accuracy of affinity calculations, particularly for populations that displace water molecules in the binding pocket. We have also been working on general constant pH method and the protein conformation FEP, and those methods will dramatically improve our capacity to deal with targets with complex formulation tautomeric states and flexible binding pocket. At the same time, we have also been working on absolute protein binding affinity FEP. And by rescoring the top compounds by inverse screen by ABFEP, the hit rate can be dramatically improved. With this, I'd like to acknowledge a large number of people who have contributed to the projects mentioned. And thank you all for your attention.
Thank you. I think Dr. Lingle is still not available with us. So it would be great if you could write down your questions in the chat box and we'll clarify with the speaker and we will address that.